Well, good morning, everybody. A familiar theme that I'm sure we'll all be discussing these few days is it's great to be back. And certainly great for me, coming from Australia, which was one of the countries that had the harshest lockdown restrictions. We spent several months under curfew and some very significant impacts upon our life. Fortunately, a low incidence of COVID-19 infections across the country, but nonetheless a significant impact to our social well-being. With that introduction, I'm Ross Macdonald, the Chief Executive Officer of Cynata Therapeutics, an Australian next-generation stem cell company. First of all, the forward-looking statements here, please do direct any inquiries through our website and the ASX, the Australian Stock Exchange trading platform. Let me open with a few comments about the company and our technology. We are developing a unique manufacturing process for the production of therapeutic mesenchymal stem cells, or MSCs. This relies on the utilisation of induced pluripotent stem cells, or iPSCs, as the starting material, thereby avoiding the most significant limitation that we face with allogeneic cell therapies, and that is a reliance upon multiple donors. As I'll explain more in a moment, the FDA has given very clear guidance to the industry in their recent assessment of a BLA that was submitted for an MSC therapeutic product, and that guidance has provided us with a clear window to the FDA's thinking about the issues related to a reliance upon multi-donor-derived manufacturing strategies. In the years since we've been developing this technology, the company has assembled a strong safety and efficacy database. We now have multiple clinical trials underway, including a phase 3 clinical trial in osteoarthritis, making Cynata clearly the leading company worldwide in the development of iPSC-derived cell therapy products. Many others now are following those footsteps, but it's great that we are still very much in the leadership position in this exciting new paradigm for the production of cell therapy products. The indications that we're pursuing have large addressable markets. Many of the indications have significant unmet medical needs, and if we're successful, obviously the company will enjoy significant value upside along with our shareholders. A quick lesson, I suppose, on MSCs 101, which many of you will be well aware of, and that is the vast amount of biology that has now been assembled around how MSCs operate and their potential utility in a wide variety of clinical indications. The central role that MSCs play in the immune system has led to authors describing MSCs as the sensor and switcher of the immune system. Of course, I won't go into all of those pieces of biology on this schematic there, but the summation of that understanding of the biology of MSCs has led to over 1,000 clinical trials having been initiated using mesenchymal stem cells. The big challenge we face, however, as I indicated in my introduction, is in the manufacture, the industrialisation or the productisation of these living drugs. The conventional process that many of you will be well and truly familiar with is illustrated in the top panel of this schematic. Reliance upon multiple donors who are prepared to donate bone marrow or adipose tissue or other tissues from which the MSCs in scarce numbers are isolated and purified and then expanded in culture to produce the finished product. There are two hurdles that come immediately to mind there. First of all, the reliance upon multiple donors and the fact that every donor is a unique individual and every donor is therefore different. And the differences in properties between donations, not only from different donors, but even from the same donor at different times, has been clearly outlined in the literature. The second issue is the massive expansion that is required to produce significant quantities of these cells leads ultimately to senescence and a reduction in potency. So between those two hurdles, we have a major challenge that is unmet with the conventional process of manufacturing. And ultimately, as we all know, safety and efficacy are the key drivers, of course, of a pharmaceutical product. But a third, that is manufacture and manufacturability, is an essential requirement if a product is ever to be commercialised. In the lower panel, you'll see the Cynata process, which we call Cymerus, which, as I indicated before, relies upon the utilisation of induced pluripotent stem cells, or iPSCs, as the starting material. Given iPSC's unique property of essentially infinite expansion, we have an unlimited starting material that provides an identical starting cell from which we can then, using our proprietary process, differentiate the iPSCs through a pathway of differentiation into mesenchymal stem cells, which are then ultimately utilised as the therapeutic product. This pathway ensures a highly consistent and highly potent finished product. The FDA, as I mentioned, has provided guidance to the industry through their assessment of a BLA for an MSC product about 1.5 years ago. Ultimately, that led to a complete response letter, but the FDA's commentary on this, which is freely available, was very, very telling in that the FDA's primary concern, at least from a CMC point of view, was around the functional heterogeneity, in other words, the differences between donors that the manufacturer appeared not to be able to control in terms of an equipotent product between different donors. So again, every donor is different. So the FDA's perspective on this was, well, please show us that you've got a consistent manufacturing process, and the manufacturer was unable to do so. On the other hand, Cynata Cymerus technology, since it uses exactly the same starting material every time, ensures that we have a highly consistent and scalable product. And the potency is retained because the expansion is not at the level of the finished product, the MSC, but at the level of the precursor, the iPSC, thereby ensuring that batch-to-batch consistency that the FDA is absolutely critical about, and of course other regulators around the world similarly. This manufacturing platform has now allowed us to move forward into the clinic, and we've recently completed a phase 1 clinical trial in graft versus host disease. It was the first completed trial utilising an iPSC-derived cell therapy product. And in these patients with acute steroid-resistant graft versus host disease, there are very few options. And so it's a significant unmet medical need, fortunately a relatively small market. But as you can see, the results there were extremely good, not only, of course, for the patients and their carers, but the company and the technology platform as well, where we saw a complete response rate just over half of the patients, that is a complete resolution of symptoms, and a survival rate of nearly all of the patients at the initial endpoint assessment at 100 days, and then a very durable response, as you can see, with the 2-year follow-up of those patients, where we saw nearly 2/3 of the patients still alive, compared with standard of care, where less than 1/5 of the patients typically are alive after 2 years. This led to a front-page article in Nature Medicine magazine, the world's most prestigious journal. It's allowed us now to move forward to multiple clinical studies. In osteoarthritis, a phase 3 is underway in Australia, a gold standard clinical study, 440 patients, randomised, placebo-controlled clinical study, which will finally answer the question that many of us have debated, and that is the potential utility of MSCs in degenerative diseases like osteoarthritis, for which, of course, there is no effective treatment, only relief of the symptoms with analgesia, ultimately leading, of course, to surgery. We have a trial in respiratory distress underway that initially started in COVID patients, but we expanded that trial to include respiratory distress from other causes, and a trial which recently commenced in Australia in diabetic foot ulcers, involving a topical application of the cells via a novel polymer matrix. We expect to commence a phase 2 clinical study in the United States in graft versus host disease later this year, pending FDA approval, and other indications including CLI idiopathic pulmonary fibrosis and renal transplantation are on our list, but right now we are pretty busy with the trials that are already underway. Turning to our strategy for development of graft versus host disease, the study that I explained before, we're advancing that with our interaction with the US FDA, and again, pending FDA approval, that trial will get underway later this year. I won't go into details of the trials that are the other clinical studies that are underway. Suffice to say that, as I mentioned before, the osteoarthritis phase 3 is a large, randomised, well-controlled clinical study. And it, I think, goes to the heart of some of the questions around MSCs that have been generated from small, unpowered, uncontrolled clinical studies, especially using autologous patient-derived MSCs. This one will prove one way or the other whether MSCs are ultimately effective, in this case, in knee osteoarthritis. The respiratory distress clinical trial is underway in Australia at the moment and recruiting patients. We had expected that trial to complete enrolment late last year, but unfortunately one of the dilemmas or paradoxes around COVID was the impact that it had upon hospital management or the management of patients in hospital and the requirement for redirecting hospital resources away from clinical research to patient management. So unfortunately our recruitment slowed down, but it's now starting to get back on track. The clinical trial in diabetic foot ulcers commenced in Australia late last year, and that is a very significant unmet medical need as the epidemic of diabetes continues across the globe, particularly in developing countries. And for at least these chronic wounds, there's been little progress made in the last century in the way these wounds are treated. So again, answering a question, we hope, whether MSCs are effective in assisting in the regeneration of healthy dermal tissue. The significant market opportunities are represented by these clinical targets, particularly osteoarthritis, which is sustained primarily by pain relief and then ultimately surgery. The other indications as listed there are also attractive market opportunities. So the investors among you, these do represent exciting opportunities for the company and for our shareholders. Looking ahead at near-term catalysts and what to look for in this journey around iPSC-derived MSCs, we anticipate completion of recruitment of patients in the diabetic foot ulcer trial later this year. We also are on track to complete recruitment of the patients in the MEND respiratory failure trial this year. We hope to progress our US regulatory strategy by way of an engagement with the FDA and getting a phase 2 clinical trial underway in graft versus host disease. Other clinical opportunities in IPF and renal transplantation we'll pursue as and when resources allow. And then longer term, the completion of recruitment in the osteoarthritis clinical trial, which, as I mentioned before, is a very large study, 440 patients, so is well powered to demonstrate utility in this indication. We have a very clear commercial strategy of partnering our technology. That's why events like this are so important for companies like Cynata, where we can interact with and hopefully attract potential commercial partners. We're a small Australian biotech company with limited access to the resources that are required for global commercialisation of pharmaceutical products. So partnering is an essential element of our business and corporate strategy. A quick overview of the company. The top shareholders are listed there. Fidelity International, one of the leading investment funds worldwide. Bioscience Managers, a healthcare institutional investor based in Australia and the UK. And our strategic partner, Fujifilm, with whom we have been cooperating for the last 6 or 7 years, both in terms of sourcing the original iPS cell line from CDI, Cellular Dynamics International, which is now part of the Fujifilm Group, as well as more recently drawing on Fujifilm's ever-expanding resources in contract manufacturing where we will be working with Fujifilm in the scale-up of our production process. The company is well cashed up to execute on a number of our key short-term catalysts, and as I mentioned before, we look to informing the market further, and obviously groups such as the ARM about progress as we advance. Finally, I'd like to thank the Alliance for Regenerative Medicine, especially for getting us all back together again at these sorts of events. It is really wonderful to see everybody face to face. I'm sure we're all sick of Zoom calls, more especially given we're an Australian company where they're usually at 3 o'clock in the morning to accommodate US or European time zones. Contact details for me and the company are listed here, so if you've got any questions or concerns, please feel free to get in touch, and obviously I'll be here for the remainder of the conference this week. Thank you very much for your kind attention. Enjoy the rest of the day.